Yeah, it's time. Hello, everyone. Welcome to the first event of our Translation Neuroimaging Educational Program in the year of 2025. I'm the moderator for this event today. My name is Rianke, and I work at the Department of Psychiatry at University of British Columbia. Before introducing our guest speaker, Martin Teek, allow me 2 minutes to have a very quick introduction of this program. So, this education program is nested within Lifespan Working Group of the Enigma Consortium, which spans over 45 countries and involves over 2,000 researchers worldwide. Our program is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The directors of this program are Dr. Shalala Haas from Mount Sinai, New York City, and myself, and we do have an international advisory committee. Please consider to join our mailing list if you wish to receive a notification of our updates. A good news that I would like to share that this program has been awarded a special projects grant for the Society of Biological Psychiatry, and we will organize an educational program in this year's annual meeting in Toronto. The schedule will come out soon, so stay tuned if you'd like to join us there. We have a YouTube channel where you can find the recordings from previous events. We are both on Twitter and Blue Sky, and if you are on either platform, we would love for you to follow us. If you wish to share your work through this platform, please get in touch. We welcome any type of collaborations. I would like to briefly mention that we have an event scheduled at February 11th, and we will have Dr. Federico Raimondo to introduce his team's work on how to build machine learning models with neuroimaging data and with minimum coding skills through their Python packages, Juniper and Learn. Today, I'm very delighted to introduce Dr. Martin Teek. Martin is a new PI at the Medical University of Vienna, Austria. His main research interests are at the interface of fMRI brain mapping and TMS brain stimulation. Martin has made significant contributions to the field by using integrated TMS fMRI technique to demonstrate effective brain target engagement during the TMS stimulation. I've met Martin a few times at different conferences and was quite impressed by his knowledge and insights in this field of integration of neuroimaging and neuromodulation. I'm pretty sure you will have the same positive impression as I did after his talk today. So now please join me to welcome Dr. Teek for his wonderful talk. Now, the microphone is yours, Martin.
Yep. Thank you very much, Young, and thank you all the organizers of that nice educational program, which I think is a super important mission that we are all on, in order to we all are on a mission somehow to provide this knowledge to everyone, because I think TMS fMRI, but also other techniques that are more and more rising, help us really to improve therapy options in psychiatric populations. And today I want to use the possibility to introduce some of the technical innovations that we actually came up with over the last years, not just us, but also other groups that are working on all kind of technical aspects. But importantly, also there are more and more groups that are incorporating those innovations into clinical trials. And therefore, I also want to focus a little bit on the applications of combined TMS fMRI in psychiatric research. And I'm a new group leader at Medical University of Vienna, but many of the results I present today are actually done by Frank Padberg's groups, Daniel Keser's groups at the Ludwig Maximilian University in Munich. There's a lot of collaboration going on with Stanford Medicine, and there are also many other research groups that we are collaborating with. And I think it's really important for me also to emphasize from the beginning on that all of this is like a global effort, and we are all people who try to somehow make brain therapeutics work better in depressed individuals. First of all, I have nothing to declare. The reason why I'm doing it and why I'm continuing to be really focused on that kind of work is because depression is a really severe disorder, as many of you know. And in 20% of the participants at the global level, you will experience at least one episode of depression. And while some patients respond actually quite well to psychotherapy or to pharmacotherapy, there are many patients who actually do not respond. And I think TMS is actually maybe the most promising treatment option for those patients who do not respond to standard pharmacotherapy. But also, I think more and more people are getting like aware that TMS should actually also be provided as a first-line treatment, because it is a super safe TMS, it's super safe FDA-approved rapid-acting antidepressant. And I think it's very important to really make this available to each and every patient who might need it. The journey actually started, I think, in 2008 with the first FDA approval after a randomized controlled trial. This is a study done in Mark George group at the Medical University of South Carolina. He delivered 3,000 pulses of 10 Hertz repetitive TMS in a 4 seconds on, 26 second off paradigm. They did 5 sessions a week and then repeated this over 4 to 6 weeks. And this was linked to remission rates of 15%, which doesn't sound that high, but you also have to consider that these are all patients that did not respond to other treatments. Then 10 years later, there were some advances in between. They actually could show in Toronto, in Dan Bloomberger's group, that if you apply theta bursts, so a new pattern of brain stimulation to a participant's DLBFC, you can significantly decrease the treatment times, because you get the same number of pulses now delivered in 3 minutes that was previously applied in over than half an hour. So this reduces the burden on the patients. It reduces also the burden, of course, on the practitioner, and therefore this has been shown to be a more practical way to deliver treatment. Within the same 4 to 6 weeks of treatment, they can show that these shorter treatment sessions now also show at least non-inferiority over the standard 10 Hertz repetitive TMS treatment. Then 2 years later, Nolan Williams group at the Stanford Neuromodulation Therapy group, so brain stimulation lab in Stanford, could show that if you now apply longer forms of TBS, so instead of 3 minutes, apply actually 10 minutes of stimulation and then repeat that 10 times a day, you have much higher response rates even. What is important to note is that if you want to develop the best TMS therapy plan, there are so many things to consider, because you have to choose first an effective stimulation sequence or pattern. As mentioned before, this could be 10 Hertz strains of stimulation. It could be theta burst stimulation, but it could also be like an individualized frequency that is better for one or the other patient. There's ongoing work on that in groups that work on EG, for example. After defining what you want to apply, so the stimulation sequence or pattern, you also need to know where you actually want to apply it. So define the stimulation target. And this could be 5 centimeters anterior to the motor cortex. It could be based on some other scalp measurements, the F3 position, for example, so the electrode position, F3. And finally, the more modern approaches use an MRI-based targeting method based on the dorsolateral prefrontal cortex, anterior cingulate cortex connectivity. And after deciding what you want to apply and where to apply it, of course, you need to know how to do that. So you need to adjust the stimulation intensity to make sure to account for different excitability patterns. And the standard for that is still a certain percentage of the motor threshold, so the intensity needed to evoke a finger movement in a patient. But of course, there are alternatives, and Martin Arndt's groups is here to mention, who actually looked at the response of ECG, so that the heartbeat changes in response to DLBFC stimulation to optimize that parameter. But there's still ongoing work to really find out what is like the optimal way to define intensity needed. And of course, you would have the option to test all kinds of permutations of this in order to come up with the best combination. But this would really take years of work, because you can imagine if you permutate all those parameters, you have to run like 100 different clinical trials to find out how the actual response is best. And this has many disadvantages. First of all, many patients are then subjected to a treatment trial that probably won't work. And second, it also it takes really long to find out what are your optimal parameters. And I think TMS, fMRI, or any other method to show direct target engagement provides some shortcut into that, because after defining what you actually want in terms of brain changes, you can optimize the stimulation procedures, probably in the future, even in real time. How can MRI in principle help TMS in depression treatment? So you can use MRI acquisition before TMS that can support target definition. So in order to find out where to stimulate, you can use a neuronavigation system. And then in the most simple approach, you would just use an anatomical landmark and then define a certain subregion of the DLBFC as the target of interest. You can use fMRI after TMS in order to find out what really happened after a longer lasting treatment scheme. And you can finally use fMRI during TMS and precisely interleave TMS pulses with image acquisition in order to really find out what's going on in the brain during the simulation process. And that actually allows you for insights into the mechanism of action. And importantly, you can actually get this so nice to have target engagement. It's also important to note, and I want to emphasize this over and over again, if you don't have an MRI machine, you can still get super effective treatment options with, for example, the 5 to 7 centimeter rule. So you define your DLBFC hotspot based on where you would stimulate for the motor cortex response, and then just move 5 to 7 centimeters anterior. More advanced measures would be scalp-based beam F3. So you would try to identify exactly the EEG position of that participant. And this is also linked to quite nice response rates. But what can MRI actually do in order to help the positioning? First of all, the DLBFC itself is linked to a lot of promising cognitive functions. And this already makes it like a very prominent target. So there's actually more basic research that could show that stimulating a prefrontal cortex target actually stimulates the whole limbic system. It has been linked to mesolimbic dopamine reward circuits. The DLBFC has been consistently found to be involved in negative stimulus processing. So there's a lot of converging evidence that already shows that you should actually consider the DLBFC as maybe a really good target for treating affective disorders. Bharat Biswal has discovered the so-called resting state connectivity analysis based on signal fluctuations in the brain that are highly correlated even in the absence of a task. And that actually also allows us to individually look at differences of the brain connections. And this knowledge has been used by Mike Fox and other groups. He was probably the first to show that it's actually the anti-correlation between the subchannel ACC and the actual treatment targets that makes the best treatment success. And in this retrospective study, he first showed that actually published targets in clinical trials that show the better response rates also have the highest subchannel ACC anti-correlation. To sum that up, the DLBFC is a really good target for depression treatment. Also, if you use just scalp-based measurements, the resting state fMRI has allowed to target specific subregions of the DLBFC, and the connectivity between DLBFC and ACC is somewhat suggested as a marker for target efficiency in MDD treatments. But of course, there are also other possible target networks, and they have not been investigated that much as the subchannel ACC DLBFC axis. I want to just mention some of them. This is nicely summed up by Leanne Williams. She actually did literature research that showed that the default mode network is very often mentioned because it's linked to rumination symptoms. The salience network leads to anxious avoidance. The positive affect network, when somehow disturbed, is linked to anhedonia. And finally, the cognitive control network is probably the most interesting candidate because it also includes those key structures, the subchannel ACC as well as the dorsolateral prefrontal cortex. And within that cognitive control network, it's actually a hyperactivation of the anterior cingulate and the hypoactivity of the DLBFC that characterizes depression. So that if you compare healthy controls with patients. And that means if you have too much activation in the subchannel ACC and the ACC structures, This is actually maladaptive, and if you have too little activation in DLBFC, you're somehow missing a control region, so a top-down process that would help you regulate your emotions. There are also other approaches to come up with targets of interest. This is work done by Shan Siddiqui and others. He actually suggests different networks based on the so-called lesion approach. So he looked at the MRI scans of patients who had all kinds of different symptoms. And what he found is that it's not always the same brain region that actually leads to the same symptoms, but it's many, of course. But those actually are linked nicely by resting state connectivity. So they kind of form a network, and this also allows you to somehow define new target networks to treat also not only a certain disease like MDD, but also specific subsymptoms, or even targets the whole psychiatric patients in a more transdiagnostic way. But the most prominent target, as mentioned before, is probably still the SGCC, DLBFC anticorrelation. And Robin Cash and his group have constantly shown, also in retrospective as well as prospective studies, that it's the distance between the optimal target, so the highest anticorrelated DLBFC hotspot, and the actual simulated region that is also predictive of later treatment success. How can we translate these findings now into a real therapeutic protocol? I think the Stanford neuromodulation therapy is a nice example of that. They now use a neuroimaging-informed treatment option based on resting state connectivity, and they have remission rates of up to 79%, which compared to the most effective RTMS protocols that are about 60%, is of course really remarkable. And that also led to really fast FDA clearance of this protocol. As mentioned before, they now use an individualized target based on resting state connectivity, but there are other parameters that are actually changed. They use an accelerated space delivery of high-dose theta burst stimulation. You can see on the right side that this is the treatment scheme. So the patients have to come in 5 days in a row, and they are stimulated over the whole day. So they have 10 minutes of stimulation followed by 50 milliseconds of rest. This seems to be like the perfect timing to get really nice effects in there, and also uses the so-called spaced learning theorem. And you repeat this actually 10 times a day. So the patients are really in there 10 hours a day. And after 5 consecutive days of this treatment, they did deliver 90,000 pulses. So this is exactly the same number of pulses that would be delivered in the conventional 6 weeks therapy plan. And they could do all of that within 5 days, which has many advantages because, of course, the patients respond faster, which gets even more important if you talk about suicide prevention. And of course, this also has some disadvantages because it's a logistical challenge to really have like multiple patients in the clinics that get 10 of these treatment sessions a day. To find out what is actually happening during such a treatment scheme, fMRI acquisitions after TMS the key. So you can, as Nolan Williams' team did here, show, for example, that if you compare a patient before that therapy of theta burst stimulation, they show a certain level of anticorrelation between the DLBFC and the subchannel ACC. But this anticorrelation is even higher increased if you stimulate them with the theta burst protocol. And then also these changes are somewhat predictive of the symptom severity changes. Another example of using fMRI after TMS is a study that we did in Vienna a while ago. We here included healthy controls just to find out more the mechanism of action. And we did acquire resting state scans once before and then twice after the RTMS stimulation. And we did that in a real and in a sham condition. For sham, we stimulated over vertex, and for real, over the left dorsolateral prefrontal cortex. And what we did for analysis to find out in a more unbiased way what is really going on in the brain, we actually based our analysis on a normative data set by Bharat Biswal. He actually identified using independent component analysis the 20 most stable networks. So normally, if you take those seeds and run it in healthy controls over and over again, within a day, you shouldn't see any changes in there that are significant. So we thought if TMS really has a strong effect, we would still see a change in one of these networks, and we actually found exactly that. So if we stimulate participants in a real condition, 19 networks stay completely stable, and the one specific network that shows alterations also includes the stimulation site, the left dorsolateral frontal cortex. And the changes we find with that network component are, you might have guessed it, the anterior cingulate connectivity change in there. And importantly, if you compare these findings of what happens after a single session of TMS with the differences we get when looking at people who are remitted from depressed disorder to people who are still acutely depressed, we find that it's actually exactly the same network that shows alterations, and it's also a similar connectivity change. What is also noteworthy in here is that, first of all, in the sham condition, we did not get any significant effects in that, which is good. And second, there is quite some variability in the response. So we need 60 subjects to actually show a significant effect in there, which also means that not each and every individual in our sample, in our healthy control sample, did even show that network effect. And I think the reason for it is that the challenge is still a little bit unclear. We don't know exactly where to stimulate and how accurately we can stimulate the target, how strong we should stimulate, and how we individualize the dose, and also how fast or which frequency. And there are many other parameters that I will come to in a bit. I think that TMS fMRI is a way to optimize all those parameters. And measuring fMRI during TMS has a lot of advantages also over the offline approaches where you would measure after treatment, because while they actually provide more insights into the neuroplastic effects, with fMRI during TMS, we can really show real-time target engagement, and we can investigate the real mechanism of action because we know what is really going on inside the brain while the TMS pulses are applied. This is like a scheme of how that usually works. We have a TMS coil, we have an MR imaging coil, which is there to receive the signal from the brain. And when combining TMS and fMRI, there are a couple of technical considerations that you need to consider. First of all, everything you want to use in an MRI machine, of course, needs to be of non-ferromagnetic material. There is, for example, a McVenture coil that consists of copper windings, and they're in a very rigid plastic housing. Also because they need to withstand a lot of mechanical stress, because in an MRI machine, you have, for example, 3 Tesla magnetic field that is static in there, and the TMS pulses are of around like 1 Tesla, and therefore you have like 2 magnetic fields that are working somehow against each other. So this just leads to a lot of stress on the coil housing, and also leads to vibrations that you can somehow help a little bit to mitigate by using a proper coil holder. This is about safety. Then when talking about artifacts, things to consider are static artifacts due to the presence of the coil. As already mentioned, it's usually made of a lot of copper windings. And if you have like copper windings between your head and the receiving element, so the MR coil, then of course you're somehow blocking the whole signal you would otherwise want to measure from your participants, which is of course suboptimal. So you have to consider the right RF coil design choice, and people were becoming super creative regarding that, using all kind of flexible and surface coils. I will show some examples a little bit later. After dealing with the static artifacts, dynamic artifacts are introduced usually because of RF coil that might propagate from the TMS cable, because you could think of the TMS coil also acting a little bit like an antenna that is leading from inside the scanner to outside the scanner. And if you connect it then to your stimulation machine that is usually outside the scanner, you get like all the radio frequency noise that is in that room, you get like transferred directly into your MRI machine. So this is something you want to avoid. Therefore, you normally use a filter. Then, of course, there are TMS-MR interactions that we can actually only avoid by precisely programming our TMS pulses to happen in specific phases of image acquisition. This is something I want to show here. So as some of you might know, the MRI data is not acquired in image space, but in case base. You don't need to understand that if not, but in general, specific times during your acquired slices correspond to specific frequencies in your images and not like points in your picture. So that means if we stimulate a little bit too early, and that is 35 milliseconds after the image acquisition of one imaging slice. This is a phantom that is spherical, so we have like, we should see like a full circle. If we stimulate now 35 milliseconds after the image acquisition of one of the slices has happened, then we can see there is a large dropout that makes it impossible to even see what this object would look like. If we stimulate after that center of K-space, so 40 milliseconds after the onset of image acquisition, we did record the whole center of K-space, so therefore the shape of the object is somewhat clear, but we have high frequency artifacts in there, which you can see as this checkerboard patterns here. And importantly, if you really wait 60 milliseconds, so if you get the timing right, you stimulate exactly after the whole slice acquisition has been done and before the next slice acquisition has been started. This is actually pretty straightforward, but usually in each and every scanner site, people need to really fine-tune that to make sure that their imaging sequences really are not affected by TMS stimulation. There are multiple ways to deal with this problem now. First of all, you could just ignore what I just said and stimulate whenever you want, and then you would get these artifacts in certain slices, and you can use post-processing to just interpolate them. But of course, this is not the most elegant way. Another option is to place TMS pulses within acquisition gaps, so you acquire a whole brain image, and then you wait for a couple of milliseconds, and in there you can place a pulse or even multiple pulses. And then finally, the most advanced way is by really precisely timing image acquisition with TMS pulses. You can precisely place your TMS pulses to happen exactly between acquired slices. Another challenge that I've talked about is the AF choice. My colleague Lucia Navarro-Lara worked with me in Vienna on this. It's a 7-channel receive array that actually gives us really nice signal at the stimulation site, so close to where these 7 coil elements are positioned. And based on that setup, what we have here in Vienna is like a TMS coil that is mounted on top of one of these radio frequencies coils. And then we have a second radio frequency coil on the contralateral side of the brain. And then to position the TMS fMRI coil array at the right side, we use a neural navigation camera that does localize the TMS coil position as well as the subject that actually is wearing a customized mouse mount with a tracker on it. So we can ensure that the subjects are stimulated at the right side, even if they are inside the MRI machine. Using this setup, one of the first things we did is we positioned the TMS coil on exactly the same target that was used in the other study with resting state connectivity that I introduced before. And we applied triplets of 10 Hertz pulses followed by 30 seconds of rest. And those pulses were also applied at different stimulation intensities. If we look at the results in the whole group of 14 subjects, we can see that acute TMS on the DLBFC does not only lead to a bold response at the stimulation site, but also at the contralateral site and in many other cortical and subcortical regions of something that already looks quite like a nice brain network. And if we then compare it to the resting state connectivity study I have introduced before, we can see that those activation patterns, regions that show a direct response to TMS stimulation, also co-localize to that network. So that means those neurons that fire together over and over again, they also wire together. So we get changes in exactly that network. And this network also shows high similarity with the state-of-the-art depression treatment network, the SGCC anticoagulation. And we also found some dose effects in there. So if we compare 110% with 80% of motor threshold stimulation, you can see there's a higher engagement of the subchannel ACC. And this is also found in other groups. For example, Fink could show that there is like a direct SGC engagement when stimulating the left DLBFC. But what you can see nicely in this figure is that he was not able to capture that with each and every participant. And this could be related to technical reasons, but it could also be part of the variability in treatment scheme. And I think therefore it's also important to really record all our technical parameters, but also the stimulation side, for example, to later find out how much of the variance we observe in those experiments is really part of the real individual variability in treatment response. And speaking of that, we did a study where we also looked at how do different doses actually affect the response, not only locally at the simulation site, but in the whole network. And you can see here, at least in these unthresholded maps, that there is a certain degree of dose dependency. So that left DLBFC over the whole group shows higher bone response if you stimulate stronger, but the same is also true for other parts of the network. And if we, however, look at individual results, it's also important to note that some participants did actually not respond to lower stimulation intensities, while others actually have a nice linear dose-response relationship. And we also looked at the linearity at the whole brain level. So this is a voxel-wise regression analysis, linear regression analysis, where we look at which of the brain regions show the highest amount of linear dose response. So which responds stronger if you stimulate with stronger intensities. And an important candidate to highlight here again is, of course, the subchannel ACC. So especially in the subchannel ACC, it seems to really play a role how strong you stimulate. Everything I've shown so far is limited by a couple of factors because everything is done in healthy participants. We don't know how that really compares to patients. Second, the typical stimulation procedures just include some single pulses or some limited sparse pulses. And we don't know how that really compares to a real therapeutic protocol where usually thousands of pulses are applied in an individual. However, the group of Desmond Otis at the University of Pennsylvania could recently show that single pulses applied to depressed patients give you a response in the subchannel ACC that is also predictive to a certain extent to the treatment response. And of course, this is quite promising because it could help to find out before the actual treatment if they would respond or not. Still, there's still a limitation because those kind of procedures do not really compare to what is happening during a theta burst protocol. That's why during my research stay in Stanford with Nolan Williams, we had a collaborative project with the Ludwig Maximilian University in Munich. This is with Daniel Kieser and Frank Padberg's groups and was funded by the Bavarian Californian Tech Innovation Grant. And we first had to set up this theta burst fMRI setup in Stanford. And since they had a GE scanner, we couldn't use the recently built Siemens coil. So what we here used is a recent GE innovation. It's a blanket-like air coil. The nice thing about it, you have here 21 channels that are placed inside a really pillow-like flexible structure. And they are super thin and flexible and stable. And you can actually use this to come up with what we call the Stanford burrito approach, because you kind of wrap your head now inside this coil. It's not as comfortable as it looks like. And the nice thing is that you get a really high SNR because your imaging arrays are really as close as they can get to your participant's head. And the images you get here on the SNR level, so the signal to noise ratio in these setups is actually more comparable to a standard manufacturer's head coil than the older TMS and fMRI approaches. And after making sure to have a proper imaging setup to avoid artifacts and get nice signal, we had to play a little bit around with sequence parameters. So we programmed our own sequence that would send slice triggers. So after each slice acquisition, there was a trigger sent at the with the exact timing delay to make sure we can actually stimulate with a theta burst protocol during image acquisition. So we set the repetition rate, so the time it takes to acquire one whole brain scan, to 2 seconds, and then acquired images in 10 image acquisition slabs. So this is always 4 slices that are recorded at the same time. And the reason why we chose this is not necessarily that it gives us the best possible images, But this actually allows us to place triplets of 50 Hertz between those individually acquired slabs. And that exactly resembles a theta burst protocol. So we now have the whole brain imaged in 10 of these slabs. And we also applied 10 bursts of 50 Hertz within those 2-second trains. And then if we follow that by 88 seconds of rest, then we have a protocol that is exactly the same that you would use outside the scanner to treat patients, like it is done in the same treatment, for example. So we were quite happy that this actually worked and also gave us really high quality API images that also translate nicely into activation changes. On the right side, you can see the individual activation changes in a single individual. And this is highly significant, even if the data is unsmooth. So we get quite nice details here. As mentioned before, other groups have also developed all kind of different creative setups to combine TMS, RF imaging arrays with the TMS stimulation, and they also developed different coil holders. And this is an ongoing endeavor to really find solutions for all kind of different problems. And it's nicely summed up recently by Alex Vulgar and others in a consensus paper that you can look up. And I want to emphasize here that there are many different approaches that do have their advantages and disadvantages. Some of them have probably a better SNR at certain sites, while others are just much more comfortable. It's also something that should be considered in those experiments, that TMS fMRI is not always like super nice, and it might actually be important to also consider patient compliance. As an example, I want to just compare two of the setups that I talked before. There is the protocol we are using in Stanford, the so-called burrito approach, which is based on the GE air coil with the 21 channels. And we can see that this gives a really nice homogeneous SNR throughout the whole brain. We also get a nice signal here in the subchannel ACC and in both cortical and subcortical structures. In general, this is similar to what you would get from a standard head coil. The nice thing about the sandwich approach that was developed in Vienna and is now also used, for example, in the Munich group or in Georg Kranz group in Hong Kong. The nice thing about this approach is that you have much higher signal to noise ratio here at the stimulation site. So if you are looking for a local dose response, you're probably better with similar surface coils. There are also other groups, FAS1, for example, in Toronto, who have worked on similar approaches. So those surface coils are really good if you want to get the signal at the stimulation site. For more homogeneous signals in the subcortical structures, maybe those flexible coils are better. And Lucia Navarro is currently working on a cap that you can wear that I think will lead to even better signals. Back to the sandwich approach that we used now in the group of Frank Pattberg and Daniel Kieser at the Ludwig Maximilian University in Munich. We also implemented here a theta burst protocol inside the MRI. So again, we set the repetition rate, so the time it takes to acquire one whole brain image to 2 seconds, place these 10 bursts of 3 pulses of 50 Hertz in this train. And then using this setup, what here Cayenne and her team could show is that if applied in 20 healthy participants, we can map the direct response. You can see in this group activation map that is actually quite some spread around the dorsolateral prefrontal cortex. So this is probably less precisely focused on a single spot than in other activation maps I've shown before. And this is caused probably by the F3 targeting method. So instead of using like a specific brain coordinate, we were actually using here the F3 electrode position. And this could be that this does not 100% align with the participants' same brain structures. But still, this seems to be a super highly effective protocol. And if you applied that in a bipolar patient, this is the first patient that got a therapeutic protocol inside an MRI machine. And the nice thing about it is that he showed really directly a target engagement in the subchannel ACC. So there was a strong modulation of this structure, acutely observable while he was getting a therapeutic session of theta burst stimulation. Cayenne also looked at those effects. So she could here somehow also confirm what we did in Vienna. So we see that for 40% of motor threshold stimulation on the left DLBFC, there is activation observable, especially around the somatosensory regions and auditory regions. And there's also some effects in the contralateral DLBFC, but to get a nice signal in the left dorsolateral prefrontal cortex, you need 80% of the stimulation intensity of the motor threshold. Now that we have all kinds of setups that allow us to measure activation changes during therapeutic protocols, we can now actually use those activation maps and then correlate them with other clinical parameters that we record after a treatment session. And I think this will be the key for future progress, because it now allows us to correlate individually evoked bold responses in subjects with the later remission, or with clinical scores, or with specific patient-specific variables. And then we can find out, is it one or the other region that is really predictive of the treatment success? And this would allow us then to fine-tune all kinds of parameters that would improve this response, especially in this before set target region. So, and parameters, I've mentioned many of them already, that we could improve or that we could individualize stimulation intensity, the protocol, the frequency, wave shape, coil orientation, but also many other factors. And given the plethora of questions we have here, I think it's really nice to have also really many groups working on all of that. So there is, for example, the pioneers in TMS fMRI, Mark George group in South Carolina, who pioneered TMS fMRI and also EG at the same time. Whenever I came up with a new idea, I actually looked at PubMed and found that Mark George published that years ago. So they really are at the foundation of TMS fMRI, and many of the other research groups are building on their first experiments. Also, Lisa McTeague is currently working a lot on patient applications of more modern TMS fMRI setups. TMS fMRI EEG also has been improved a lot by the group of Alex Sack in Maastricht, and they looked at EEG-based state dependency effects, also with Theresa Schumann. My group in Vienna is continuing to work on sequences that allow for all kinds of different therapeutic protocols. And we currently also are working on chronometric approaches. So that means we are now precisely TMS pulses to occur during specific cognitive sub-process. And University of Pennsylvania, Desmond K. Oates group did show, as mentioned before, that the SGACC response can predict treatment success. In Stanford, there was the first implementation of a therapeutic theta burst simulation on a GE scanner. Then the Moni group was the first to run that in a real patient. And a similar setup now has been also implemented in Hong Kong in Georg Kranz's group. He is looking at dose response effects with theta burst fMRI, as well as also theta burst functional near infrared spectroscopy. In Toronto, Maria Vasiladi is now working with Sean Nestor to run a different approach, again, based on flexible Siemens coils, to take a look at theta burst effects in treatment-resistant depression. And Vancouver also plans to implement the TMS fMRI setup in a clinical study. And finally, I want to give a little outlook and talk a little bit about what my lab is currently doing. We are now focused on state dependency of TMS. Why is state important? Because it is of clinical relevance. It has, for example, been shown that if a working memory task is performed during TMS protocols, this leads to higher success in terms of depression score reduction. While if patients are actually asked to focus on negative thoughts, this seems to hinder the response to otherwise effective TMS protocols. So we wanted to know why this is the case, or what is like the key to working memory processes interacting with TMS pulses, and that might link to target engagement or like differences in the response to TMS. And what we did is a planning session first, where we acquired anatomical scans in MBAC task and then resting state connectivity. The resting state connectivity was used to target the SGACC DLBFC hotspot, similar to like Robin Cash and others are doing it. So we use the standard targeting depression treatment, the SGCC anticorrelated DBC hotspot. We position our TMS fMRI coil array on there, and then we run 3 different tasks. One is stimulation during rest. So participants are just getting triplets of 10 Hertz at random times. And then we have an easy task, a zero-back task where people are asked to press a button whenever they see the letter X. And then a more complex task, the 2-back condition, where people are asked to press a button whenever the letter they currently see matches the letter they saw 2 trials earlier. And for both of these conditions, so for the easy task and the complex task, we combined it with 2 different modulation or stimulation conditions. And one is effective and one is ineffective. And what I mean by that is that in the ineffective condition, we placed a triplet of 10 Hertz, 250 milliseconds before the onset of the letter, which means for the participant in their experience, they actually think that they are stimulated while they see the letter X. But in reality, the stimulation actually occurs slightly before they could even visually process that stimulus. And the nice thing about the brain, which makes it a nice organ to work with for chronometric TMS fMRI, is that it takes certain time to process stimuli in specific regions of the brain. So before the information you get from that letter is actually encoded in the left dorsolateral prefrontal cortex, it needs at least 200 milliseconds. So we placed in the effective condition the TMS pulses to occur 150 milliseconds after stimulus onset. So that means in the effective condition, we are sure to somehow interact with the ongoing task processing of matching those letters. While in the ineffective condition, we might get some other effects that are maybe more priming, but we don't directly interact with that effect. And nicely, if we compare now effectively timed pulses versus ineffectively timed pulses in 2-back versus 0-back, so also contrasting for the control condition, we can see that actually when stimulating at the right spot at the right time, we get stronger engagement of the left dorsolateral prefrontal cortex and the subgenual ACC. And what is happening, we are downregulating the SGACC and upregulating the DLBFC. And as mentioned before, the cognitive control network was found to be changed in depressed patients, and they showed too little activation in the DLBFC and too much in the SGACC. So we can somehow reverse this connectivity pattern even better if we take timing into account. To sum this all up, as mentioned before, TMS is a very effective depression treatment option. And I want to emphasize that even if you don't have an MRI machine, but have a TMS stimulator and the patient population waiting for the treatment, I think you should start to treat them because it's a better alternative than most other pharmaceuticals that you could get. But if you have an MRI machine, this can really help to advance and inform brain stimulation procedures and leads to higher success. You can use neuroimaging before TMS to define the target. You can use it after TMS to understand better what is happening in terms of lasting network changes. And finally, we can do it during TMS to really show direct target engagement, broad parameter choices. We have, not only we, but also other groups have shown that the TMS efficiency depends not only on technical factors like stimulation intensity, but also on inter-individual factors, for example, different groups or sex differences. And we also show state dependencies in there. So also within a subject, the response to TMS on the specific region might differ. And finally, if we find a way to somehow build a closed loop with interleaved TMS fMRI, this could in the future enable us to have a session of TMS fMRI, maybe a therapeutic protocol with different intensities, frequencies, and so on, that would allow us to choose the optimal parameters in real time. And with this, I want to thank you all for your attention. And if you have any questions, feel free to ask them.